This is a diagram of the cell cycle. As you can see, the cell goes through 4 main phases in its lifetime. Growth 1, where a cell grows in size and duplicates all of its organelles. Synthesis, where a cell duplicates its genetic material. Growth 2, where a cell continues to increase in size and checks itself for errors that might hurt any future daughter cells. And mitosis paired with cytokinesis, where a cell splits apart its genetic material and divides into 2 identical daughter cells. Growth 1, synthesis, and growth 2 are all part of interphase, when a cell is not actively dividing and is performing all of its normal functions. Let's take a closer look at the different stages of mitosis. This is prophase, the first stage of mitosis. This cell looks very similar to a cell in interphase. However, the duplicated genetic material is no longer in the long, stringy form of chromatin, but rather the shorter and more easily visible form of chromatids. Note that where this diagram is labeled chromatin, it should say chromatids. The chromatids are jumbled together in the nuclear envelope. The next phase of mitosis is prometaphase. In this phase, the identical pairs of chromatids have joined together. These pairs of sister chromatids are also called duplicated chromosomes. During interphase, chromosomes are not duplicated but consist of one strand of chromatin. During prometaphase, the cell membrane begins to dissolve, and outside of it, centrioles and asters that were duplicated in growth 1 form spindle fibers between each other and move to opposite poles of the cell. This diagram is labeled prophase because prometaphase is an extension of prophase that leads into the next stage, metaphase. In metaphase, the sister chromatids float in the cytoplasm as the nuclear envelope has dissolved. Spindle fibers attach to the middle of the chromosomes, called the centromeres. The spindle fibers pull the chromosomes directly in the middle of the 2 centrioles along an imaginary plane. The next phase of mitosis is anaphase. In this phase, the spindle fibers pull the chromosomes apart, separating the sister chromatids. The sister chromatids are pulled to opposite poles of the cell to prepare for the next phase. The next and final phase of mitosis is telophase with cytokinesis. As you can see, the cell is stretching, the spindle apparatus made of the centrioles, asters, and spindle fibers has dissolved, and the separated sister chromatids are on the opposite sides of the cell. The cytoplasm begins to separate at the cleavage furrow, and the nuclear envelope is reforming around the genetic material. Soon there will be 2 new daughter cells, each with full set of chromosomes identical to those of the original parent cell. Then, the cell cycle starts all over again. Thank you for watching.